Okay, everyone, this is the second part of the video lecture for Chapter 15. If you remember at the end of the first part of this, we were talking about competition between males for access to females and how males have to compete more intensively with each other for access to the opposite sex typically than females do. So now let's talk about, to head off the second part of this video lecture, contests between males and between sperm. So males of a lot of species possess ornaments or bright coloration, allowing them to compete for females through visual displays. We kind of got into that a little bit in the last chapter, or the last video lecture for this chapter. And many of these ornaments that we see are also actually used as weapons. Think about tusks and horns and that sort of thing, antlers, for example. And these are used during combat with competing males. Plumage patterns and songs in male birds often mark individuals so as to establish dominance hierarchy in the population of males in that species. So here we see, in this figure, just an example of some of the what can be in some cases absurd ornamentation that males often have. Here you see just some pencil line drawings of various species, some of which are extinct, some of which are still around. But for example, you see the narwhal tusk here, right? That's an extant species still around. See the same thing in some lizards and geckos and that sort of thing. You see trilobites, which are all now extinct. Some of the males had very large horns. You see that in some modern fish. Look at some of these large mammals that existed in the Pleistocene prior to their extinction. Look at dinosaurs, for example, like the ceratopsians. So lots of examples of these sort of, extreme male ornaments typically used for competition, for access to females. So selection for large male ornaments such as these can result in an evolutionary arms race in which ever larger ornamentation evolves because males possessing these features leave more offspring, males of which also have alleles for those features too. So, but some arms races obviously can't go on forever because conspicuous male ornamentation has a price. It's expensive to produce, for one, and maintain as well, but it also may actually ultimately reduce survivability. So there's going to be an equilibrium that's reached at some point between sexual selection for these extreme ornaments and ecologically driven selection for the reduction of these ornaments. So Darwin noted that the equilibrium value of an ornament, so its size, its brightness, whatever, is likely to be greater than expected if only ecological selection were operating. He was suggesting there that the reason why we do have these large ornaments is because sexual selection acts to sort of amplify them and make them larger than they would be, or brighter, or more gaudy, or whatever, than they would be if we only had ecological selection operating. He observed that females and non-breeding males of many species are often drab in coloration and lack conspicuous ornamentation. That suggests that bright coloration and conspicuousness are disadvantages from an ecological perspective. Now, male competition doesn't only occur prior to copulation with a female. It certainly does, but that isn't where it always ends. A male is often capable of reducing the likelihood that sperm of another male will fertilize a female's eggs once he has copulated with her. Males of many species actually guard their females and prevent interaction with other males. A male may physically hold on to a female for as long as she produces eggs, thereby preventing other males from fertilizing those eggs. You see this in some invertebrates like crustaceans. You see it in frogs, for example. In frogs, there's a mating embrace called amplexus, where the male grabs the female from behind and basically covers her eggs with sperm as she releases them. And as long as he's the one there, he's the only one capable of fertilizing those eggs. You also see this kind of thing in some insects as well. The seminal fluid of a male may make a female less attractive to another male. It may reduce her sexual receptivity, or it may form even a vaginal copulatory plug that physically prevents another male sperm from reaching her eggs. You see this in species with internal fertilization. So you see it in some snakes, for example, fruit flies, and various others. Now, competition may continue after multiple males copulate the single female. This is known as sperm competition. Okay, so we have something called sperm competition. And this is where the sperm, technically, of two different males, let's say, are competing with each other for access to the female's eggs. For example, a male's genitalia may have a morphology allowing removal of a previous male sperm from the female reproductive tract. This is the male reproductive organ of a damselfly. I forget what species, but that's the reproductive organ. And you can see the odd shape to it, almost shaped like a hook or a scoop. And you see this is actually sperm from another male that was removed by this male from the female's reproductive tract. That's an example where the male's genitalia may have a morphology that allows it to actually, pull out sperm from a previous male's copulation with the female. Okay, so when sperm from multiple males have an opportunity to fertilize the eggs of a single female, a male that produces a larger number of sperm is obviously going to be more likely to sire her offspring. It's at that point really just a numbers game. Who produces more sperm, or whoever does, is going to have a higher probability of actually being the one to fertilize those eggs. So this type of sperm competition explains why males of polygamous primates, for example, have larger testes than males of monogamous species. And that's what you see here. So if you look at body weight in kilograms and you look at testes weight, so we have a ratio then, a relationship between the weight of the testes and the weight of the body, and you see that the black circles represent monogamous species. So first thing is that as body weight gets larger or increases, we have larger testes size. That makes sense. There's a positive correlation there. We see the same thing in polygamous species. What you want to notice, though, is that all of the polygamous species, for a given body weight, their sperm, or their testes rather, are larger than those of monogamous species. All right. So this suggests that the polygamous species, pound for pound, I guess, can actually make more sperm than monogamous species. And that would be an advantage in a polygamous society where you have multiple males mating with a single female, let's say. All right. So this brings us to a concept known as sperm precedence. Okay, sperm precedence. This occurs when most of the eggs of a single female are fertilized by only one of multiple males she is actually mated with. So she may mate with multiple males, but her eggs are fertilized by most of her eggs, let's say, are fertilized by only one of those males. So genetic variation among males then affects sperm precedence. Sperm of some males are better at displacing sperm of other males, and on the flip side, sperm of some males are better at resisting being displaced by sperm of other males. So this is thought to actually explain, in large measure, the huge variation in size and morphology of sperm among different species. Human sperm, obviously, and most mammal sperm are extremely small, very, very small compared to the egg. But you have, in some cases, extreme sizes in sperm. There's a species of fruit fly, for example, that has sperm that are upwards of 23 millimeters in length. Now, in diameter, they're still microscopic, but they are 23 millimeters long. Okay. Now let's shift gears a little bit and talk about sexual selection by mate choice. So we talked about the fact that, earlier, a female could theoretically have all of her eggs fertilized by a single male. And again, if she makes a bad choice in terms of a mate, that could dramatically impact her lifetime reproductive success in terms of her offspring's fitness as they go on and develop and that sort of thing. We talked about the fact that a male can produce large numbers of sperm for multiple years and spread those sperm far and wide, basically. And that means that males are generally going to compete for access to females because females are the choosier species because they have choosier sex because they have a lot more to lose by making a bad mate choice. So males of many species have to compete to be chosen by a female. Mate choice, again, usually by females, is therefore a very important selective agent in the evolution of male reproductive features. Such features are often elaborate or ornate morphological characters, as we've seen. They can be complex behaviors, and they're often ecologically disadvantageous as well. So on one hand, from a sexual selection standpoint, they can be very advantageous, but on the other hand, from an ecological selection standpoint, they can be very disadvantageous to the male. Great example of this from the Hawaiian Islands. there's a Hawaiian cricket, Teleogryllus oceanicus, the males of which call to attract females by striking their wings together. The male has a file-like structure on one wing and a plate on the other wing, and by rubbing that file-like structure across that plate, it makes what's called a stridulation noise. That's the cricket sound, right? They do this to call females. Now, once a female arrives, the male then initiates a courtship song to convince her to mate, again, using that stridulation process. On the island of Kauai, recently there was a fly species introduced, and this fly is a predator. It preys on crickets. And so this introduced fly species homes in on its cricket prey by listening for the male songs. And biologists have found that just in over 20 generations, the male population of crickets has evolved so that now most males no longer call. Because if you call, you're attracting a predator to yourself. And so those males that were calling were basically being selected against because they were being taken by predators before they could actually reproduce. So now we have a population of males of this cricket species whose wings are modified such that they cannot produce sound. There's a mutation that causes a morphological change, a structural change in the wing, and they can no longer produce the sound. The silent males now gather near the few remaining calling males, and they mate with the females that are arriving as a result of the calling males. But it also turns out that many females have evolved in response by no longer requiring a courtship song to initiate the mating. So they're adapting as well to the new population of males. And from a more morphological standpoint, you can see the difference here. So in panel A, you can see on that section of the wing, you have this large file-like structure. This is in a male that would make that mating call to the female. These are the ones that are declining in frequency because of the fly predator. Now, most of the males have this altered morphology where you see a much reduced file-like structure, and it's actually changed, or the location of it's changed. They physically cannot make that stridulation noise anymore. This is a great example of how selection operates, in some cases over very short time frames, to dramatically alter, in this case, reproductive behavior of a species. All right, so what can a female tell about a male based on his ornaments or other features? The answer generally is thought to be that she can tell something about his genetic quality and his potential to favorably contribute to her offspring through nutrition, resources, parental care, whatever. These are direct benefits of mate choice to the female. So, females then are selected for who are able to recognize quality males via a male feature that is correlated with his capacity as a provider. If a female can tell that a male is going to be a good provider, then she's at an advantage over females who can't make that distinction. So, female preference then selects for males with such characters. This leads us to talk about something referred to as runaway sexual selection. So, the evolution of female preference for a male trait and the evolution of the male trait become a self-reinforcing process that can snowball toward highly exaggerated male features. And the important point to grasp here is that offspring receive genes from both of their parents. So, daughters will have their mother's gene giving them preference for a certain male character, but they also have their father's gene for that character, although it's not expressed in the daughters. All right? Now, because females exhibit different preferences for male genotypes, such as phenotypic characters, or through phenotypic characters, the mating pattern produces in the next generation a correlation between the male character in the sons and the mate preference of the daughters. So alleles for female choice and the male character become associated with each other through linkage disequilibrium, as we've seen before. So mothers prefer a certain male character and pass that allele to their daughters and to their sons. The father possessing the character passes that allele on to his sons and to his daughters. We all get our DNA from both parents. So preference for the character and possession of that character end up evolving together. If a male possesses a particularly exaggerated form of a character and a female finds that exaggerated character attractive, their daughters will find it attractive and their sons will possess it. Any change in the frequency of a male character should result in a change in the frequency of female preference for it. It's exactly what we saw with the crickets. This means sons can pass along to their daughters the allele for female preference of a male character. Daughters likewise pass along to their sons the allele for the male character. So, parents pass the alleles for a male trait and preference for it to all of their offspring. This results in a self-reinforcing system, what is called the Fisherian model of runaway sexual selection after R.A. Fisher, who first proposed it. So, take a look at this figure. All right, so this can be a little bit confusing, but let's look at this top figure first. So up here we have different types of males. So male type T1 and type T2. So let's say that T1 males have a short tail, is in some species of bird, and T2 males have a long tail, all right? And here we have the probability of mating by a female of type P1 or P2. So let's say we have type P1 females, right? P1 females, because you have a peak there, prefer T1 males. They prefer males with shorter tails, all right? Now, you'll get some that will mate with T2 males, as you see over here, but not very many. Most P1 females mate with T1 males. Likewise, P2 females prefer T2 males with long tails, so they'll preferentially mate with T2 males. Now, some of them will mate with T1 males, but not very many. Most, again, mate with T2 males. All right, so what does this do then in the next generation? Well, here on the y-axis in the second graph, we have the daughters' mating preference, the daughters of these parents, okay? All right, here we have the son's trait. So notice that if we have a T1 male, okay, here, then most individuals will be from, let's say, these females, P1 females. Most males will be T1, and most females will prefer that type of male. Yeah, you'll get some P2 females who will prefer T1 males, but not that many, all right? Now, over here, we have T2 males that have a long tail. So those are most abundant from parents where the mother preferred T2 males, and the sisters, let's say, of these males will have a preference for that same type of male. That's that correlation that we talked about that results in that runaway sexual selection, okay? The idea is that if a female mates with males or prefers males who have long tails, then her sons will have long tails and her daughters will have preference for long tails. If a female prefers males with short tails, her sons will have short tails and her daughters will have preference for short tails. That's what that figure is telling you, okay? All right, so the question then is, are there actual indicators, physical, phenotypic indicators of genetic quality of a male? Turns out there might well be. If females are choosy, it stands to reason that they are so because they want, let's put that in air quotes, want to choose a high-quality male to contribute good genes to their offspring. Now, it's not like, female humans go to a bar on a Saturday night and say, ooh, who has the best genes? No, it's not like that. There are physical attributes that females are sort of, from an evolutionary perspective, cueing in on, okay, whether they realize it or not. So, again, if females are choosy, it stands to reason they are choosy because they want to choose a high-quality male, because that high-quality male will then contribute his good genes to her good genes, which will then produce offspring of high genetic quality, which should reproduce successfully and pass on the mother's genes ad infinitum. So, if this is true, then any female with the ability to detect a quality male through some phenotypic trait he may have would be at a selective advantage over females who did not have that ability. This is especially true if the male trait is actually correlated with genetic quality, or his genetic quality. In other words, the male trait should be what's called, or known as, a condition-dependent indicator of his fitness, a condition-dependent indicator of his fitness. Okay, so this phenotypic trait, whatever it is, should be a condition-dependent indicator of his genetic quality. Females can't do on-the-spot genetic testing, of course, so any physical indication of genetic quality is useful. This is known as the good genes. Oops, sorry guys. good genes hypothesis. Okay, good genes hypothesis. So is there any evidence for this hypothesis? Yes. One that we'll talk about here, the degree of coloration, and as it relates to parasite resistance in male sticklebacks. This is a type of fish. Offspring with fathers possessing bright red belly pigmentation seem to be more resistant to tapeworm infection. Turns out that carotenoid pigments responsible for red colors seem to enhance the immune system development. So this means that female sticklebacks should be attracted to bright red males because the males then have a greater immunity to tapeworm infection, and they should be able to pass that immunity on to their offspring and also therefore to her offspring. So here we have some data actually showing this. So on the x-axis here we have the redness of the father, right? How they measure that I'm not quite sure, but you can see the red belly coloration there. Here we have the offspring infected by, or the percent of offspring infected by tapeworm. And what you see is that as the redness condition of the father goes up, in other words, more carotenoid pigments, the percentage of offspring infected by tapeworm goes down quite dramatically. Okay, so that seems to be evidence suggesting that, at least in this case, that particular phenotypic feature of the father, the redness of his belly coloration, or the intensity of it, I guess I should say, might actually be a condition-dependent indicator of genetic quality. So female sticklebacks then would do well when on the hunt for a male mate to select a male who has the brightest coloration she can find. All right, that then would allow her to combine his good genes with her good genes, producing high-quality offspring who reproduce, and the more they reproduce, the more they pass on copies of her genes, as we talked about in previous chapters. All right? Okay, so that is then the end of chapter 15 on sex and reproductive success.